Just give me a minute because I just need to test a whole lot of things because I had my computer redone and it doesn't seem to be, we're all right. Okay. Okay, so I hope that, first of all, you can see the screen. I hope that I'm recording. I hope my audio is on and everything else. Yes, I think so. So good morning or good afternoon or good evening, whenever you choose to view this video. I'm actually taking a giant jump. We need to get on with our discussion about bacteria. And so I'm jumping straight from our discussion about COVID now to the start here, but I will set a small homework extra credit for you to cover just the last virus that I really wanted to discuss, which was HIV. But I think you probably already know quite a bit about it, but I'm going to set you a small extra credit to cover that section. In the meantime, we're going to move on to a discussion about bacteria, about the prokaryotes in particular. And I want to point out, first of all, remembering the viruses are completely non-living, but the prokaryotes are all very much alive. They have all of the definitions of life. They are cellular based. They have a cell membrane defining the cell. They function using DNA as the hereditary material with all of the cellular mechanisms that satisfy the central dogma. The DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes. So the prokaryotes are divided into two large domains, and these are the bacteria and the archaea. They have similarities. They're both prokaryotes. There's no nucleus, no membrane-bound organelles, etc. But they are, in fact, quite separate from one another. And they have distinct molecular features which separate them. And we believe that in evolutionary terms, they are quite different to one another. And in fact, the eukaryote cells quite probably arose more from archaeal ancestors than from bacterial ancestors. We won't discuss the archaea in great detail. We're going to spend most of our time discussing the bacteria. And even then, we're only able to skim the surface. And I'm just going to go through some of what are the most important groups, or the best known groups especially. I want to point something out to you. The bacterial classification is extremely difficult because in the old days, it was all done on morphological features and on metabolic features as well. We use clinical tests, for example, to distinguish between bacteria. But this is actually a very poor way of separating the bacterial groups, because bacteria may actually independently evolve the similar metabolic processes, which make them look somewhat similar, but then we discover that they're actually very, very different in other ways. Using the old systems, we had set up some clear-cut guidelines about how to classify bacteria. But came DNA sequencing or nucleic acid sequencing in general, and it became apparent that everything we knew about bacteria needed to be reworked. And it is difficult to slot what we know about the bacteria into the old classification systems of phylum, class, family, etc. Even the term species in bacteria doesn't mean an awful lot, because a species can exchange DNA, for example. Nonetheless, using both molecular and the old clinical tests and everything, some sort of scheme has been arrived at. And this very complex looking diagram is the result. Now, the first thing to note is that we do set up distinct groups. This one here was the Proteobacteria, This one you're going to hear quite a bit about. Into this group fall many of the gram-negative bacteria, which we are particularly interested in. This group here, we will also talk about the gram-positive bacteria, and also a little bit about cyanobacteria. The rest, I may mention some of these, but not probably in great detail. It's going to be these groups here that we're particularly interested in. But the one thing that's really important about this diagram, when you looked at it, it says this is a phylogenetic tree of the major lineages of bacteria. Now, this is based on a sequence of ribosomal RNA in the ribosomes, which is actually a very good way of distinguishing one bacterium from another. This allowed us to set up these groups, the groups that do have quite set distinction from one another. But what I want you to notice is this mess here, because this is supposed to be a phylogenetic tree. It should be reflecting ancestries and common ancestries, etc. It doesn't at all, because we cannot really discern much about the evolution of these different groups. We can distinguish the groups one from the other, but we have very little knowledge about what their origins are. So that is, in scientific terms, and in biological terms, a little bit unsatisfactory. Nonetheless, what we'll do is we'll select our groups of particular interest, and we'll discuss some of the features of them. So here they are and I'll point out to you here the Proteobacteria. That's going to be the very first group that we're going to talk about. They are divided into classes, and these each have a Greek name: alpha, beta, gamma, delta, epsilon. So we're going to discuss those groups. The next lot that we're going to talk about are going to be the bacteria, and the first of these are the firmicutes. The firmicutes are gram-positive bacteria that produce spores. So that is Bacillus and Clostridium. And then we'll also mention allied groups here that allied to the gram-positives are two strange groups. And one very important in terms of human and animal health, which are the mycoplasmas, and the other called the actinobacteria, which are common environmentally and very, very important ecologically. So we'll talk a bit about those as well. The others, we will mention some of them, cyanobacteria in particular, and then one or two of the other important genera and groups. Okay, so our first group, and really a huge and major group of bacteria, are the Proteobacteria. And the Proteobacteria they're named for the god Proteus, and Proteus could adopt many different forms and many different morphologies. So that's where they get the name from, and it describes them perfectly because the Proteobacteria are extremely diverse, and they occur in many different morphological forms, etc., etc. They're all gram-negative. Remind yourselves, please, about what constitutes the gram-negative wall, okay, the wall structures. Please go back and do that. They're also all chemoheterotrophs. That means that, like us, they depend upon already formed biological molecules for both their energy and as a source of their molecules to make their biomolecules, primarily their source of carbon. This is the largest of all of the groups of bacteria. And so it's of considerable importance, but it's also extremely complex. And it has become divided into these 5 classes, largely on the basis of molecular studies of the 16S ribosomal RNA and of the DNA sequencing as well. So those are the Proteobacteria, and we're going to start off this with the first group, the Alphaproteobacteria. And a typical example of the Alphaproteobacteria is actually one of the most abundant organisms on the whole planet. And it is this, it's a very small, extremely small marine bacterium called Phylogenetic. And the Alphaproteobacteria have one characteristic, one metabolic characteristic, and that is that they are extremely successful in low nutrient environments. The gram-negative structure, the outer membrane, and all of the associated features of the gram-negative morphology confer an advantage to bacteria in low nutrients. They are able to accumulate rare nutrients which they may need. So this little bacterium, Phylogeobacter, is one of the most abundant organisms not in the oceans, and in fact, its biomass is significant in the planet as a whole, and it is extremely important in the cycling of carbon in the oceans. So that is its principal interest. So here are some more of the Alphaproteobacteria. This one is called Azospirillum, and Azospirillum is a bacterium. It grows free living in soil. It uses nutrients excreted by plants, by plant roots, and as a result, it becomes very closely associated with plant roots. And it is one of the bacteria which is able to fix nitrogen, and it builds up nitrogen compounds in the cells, and when the cells die, those nitrogen compounds can then be contributed to the plants, etc. But in addition, Azospirillum, actually, this association with plants is very, very close one. It's a symbiosis. And they provide some very interesting substances to plants. They provide growth factors, for example. They even produce antibiotics, which are protective of the plants and of the plant roots. That is Azospirillum. You may wonder why it's called the spirillum at all, but there are things which link it to the spirilla bacteria. The next group, I haven't got a picture for you, but these are the Acetobacteriaceae. And the Acetobacteriaceae, the most important one is Acetobacter. And that is important for our, industrially for us, because Acetobacter is a strict aerobe. It's an obligate aerobe, but it converts ethanol to acetic acid. In other words, it turns wine to vinegar. That was its first discovery, that we produce vinegar from wine using Acetobacter. It is extremely important industrially because we converting ethanol into acetic acid we use acetic acid widely in industry, and quite apart from the fact that it produces the condiment of vinegar. Nonetheless, it's also very important if industrially. Okay, so now in the Alphaproteobacteria, this is what we mean by saying that there is such a huge variability, morphological and metabolic variability in the Proteobacteriaceae. This one is called Rickettsia. And Rickettsia is an extremely small bacterium, and it is an obligate intracellular parasite. What else have we learned about that was an obligate intracellular parasite? The viruses. And there are many people who believe that viruses may have evolved from something like a rickettsial cell. They're extremely small. There are a number of them, and it's here all over the world. They cause a group of diseases called the spotted fevers, and that describes them absolutely. They have produced very often a very high temperature associated with a widely dispersed rash, And the most famous one here in America is Rocky Mountain spotted fever. They are all transmitted by insect and tick bites, all of the Ricketsias. And they usually, this is the deer tick which transmits Rocky Mountain spotted fever here in the United States. And this is a typical symptom of Rocky Mountain spotted fever. Rocky Mountain spotted fever, actually, it occurs far away from the Rocky Mountains. It's almost unknown in the Rocky Mountains. I don't quite know how it got that name, but it's mostly associated with the eastern seaboard, not the Rocky Mountains themselves. And it can be an extremely dangerous disease. It's the whole life cycle of Rickettsia is really interesting. It is released from the cell as an infectious unit, but it has to be taken up by an insect vector, and it is then transmitted to another host. So it has to have that intermediate host, the tick or whatever. There are some other insects, especially elsewhere in the world, which can transmit Rickettsias. And when it infects its terminal host, this is a culture of it in chicken embryo. And you can see here's a cell here, the chicken eggs, the chicken cell, embryo cell. Here's the nucleus here. And here you can see the Ricketsias. Imagine this is just a single cell. This gives you an idea. They're hardly bigger than the mitochondria. And they're scattered throughout the cell, throughout the cytoplasm, but they also congregate in the nucleus. And they cause extreme disruption of the functioning of these cells. The Ricketsias in general are associated with a very high fever and a very bad headache, and then this outbreak of the rash as well. And they are commonly called tick bite fevers, apart from Rocky Mountain spotted fever. A cause of disease widespread throughout the world, and several different kinds of the Ricketsias as well. They undergo a strange, we won't go into all of the details, they undergo a complex life cycle within the cell in order to produce these infectious particles. The next members of the Alphaproteobacteria that we'll talk about are these very strange morphology. They're again bacteria which are found in low nutrient environments, in fresh and marine systems. And they have this strange morphology. The two genera to remember are Caulobacter and Hyphomicrobium. And probably Caulobacter is the better known of them. So they're a flagellated cell, but this flagellum, and in common with bacterial flagella for other bacteria as well, can be regressed and regrown. And when it has the flagellum, the cell is free swimming and widely dispersed. And but when it approaches a suitable surface that it can settle on, the flagellum regresses, and instead they grow a thick stalk, and the stalk attaches. Once the stalk is attached, then only then does the cell enter a division phase. And they divide by budding, and they bud off a flagellated cell. So in actual fact, although they show it here as being almost equal in size, often this cell budding off here is very much smaller. But it has a flagellum, and it now produces a free swimming form, which can disperse and then settle elsewhere. Our next group are probably, in terms of global ecology, amongst the most important of all of the bacteria. These are the bacteria which are primarily responsible for fixing nitrogen in association with plants, and especially with a group of plants which we call the legumes. That is the peas, beans, all of those sorts of plants. The process of nitrogen fixation is important because free nitrogen in our atmosphere is extremely abundant. So that is N2. It occurs in the dinitrogen form. And N2 is practically inert. It's extremely difficult to get it to react with anything at all. And it is certainly biologically useless. We breathe in nitrogen the whole time. All we do, breathe it out again, because it does absolutely nothing. It is completely non-reactive. So that is a problem, because much of biology depends, of course, upon the construction of nitrogenous compounds, nucleic acids, the proteins. These are all rich in nitrogen. And there are other substances, in biological substances as well, that use nitrogen. So somehow or other, the atmospheric nitrogen, N2, has got to be converted into a biologically useful form. The simplest of the biologically useful forms is ammonia, or NH4 plus, if it's the ionic form, NH4 plus the ammonium ion. That is the simplest form, and that is the responsibility entirely of bacteria to do that. It is only the bacteria which are able to take atmospheric nitrogen and convert it into a biologically useful form. So the bacteria that can do this lie at the basis of the whole biological cycling of nitrogen through the biosphere. And these Alphaproteobacteria, especially these two, Rhizobium, and then a relatively recently described species, genus rather, Bradyrhizobium. They're very, very similar to one another. And together they form a group called the Rhizobia. These bacteria are free living in soil. They can be free living, but when they encounter plants with certain molecular receptors on their root hairs, they're able to fix to those root hairs and induce the root hairs to form small nodules. You can see them here. And within those nodules, these bacteria are able to fix nitrogen into biologically useful compounds. It's a very complex process, but they do in fact provide fixed nitrogen, not only for themselves, because they need nitrogen for their proteins and nucleic acids and everything, but they are actually able to supply nitrogen to the plant to which they are attached. So these nodules here are typical of the legumes. If you pull up bean or pea plant, you will find them quite easily on the roots, and they're called the root nodules. And they're formed from root hair tissue surrounding clumps of the bacteria. Here you can see bacteria here, and surrounding them is plant tissue, forming this very intimate association. So the bacteria are now being sheltered, and they're being provided with molecules which they can use, especially with sugars, substances like that come to them from the plant tissues. They respire those for energy and for useful molecules, of course. And they use the energy to fix nitrogen, which is a very energetically demanding process. But they fix first into ammonia, which they can use, and which the plants, if there's excess ammonia, then the plants can take it up and use it as well. But most of the nitrogen is provided to the plant in the form of the amino acid asparagine. And that is easily absorbed by the plant. And there are actually specific transport mechanisms from the root nodules into the plant tissues to transport that. This is our major source of fixation of nitrogen. So in the old days, in order to fertilize fields, you either use manure, which contained abundant proteins and things which were being degraded by bacteria and liberated nitrogen into usable nitrogen into the soils, or else you could grow cover crops of legumes, and then at the end of the season, when the plants were mature and their roots were full of these nodules and everything, then they would plow the plants into the soil and allow other bacteria to decay the plants and liberate all the nitrogenous compounds into the soil to fertilize the next crop, whatever it was that they were making. So that's one system of nitrogen fixation. Now here's another one which has proved to be very useful. And this is actually a pathogen. It's not a pathogen which causes crop failures or anything like that, but it occurs quite regularly. It is a bacterium called Agrobacterium, and it occurs free living in the soil, but it forms an association with plants as a pathogen, and it causes a tumor to develop. And here it is here. But they do this in an extremely interesting way. And that is that Agrobacterium has a plasmid, which it can insert, which it can produce, which gets taken up by the plant cells. So it's effectively injecting a plasmid into the plant cells. And as a result, the plant cells are transformed and start to form this tumorous system, which on an individual basis can be lethal to a plant. But in terms of genetic engineering, this is hugely useful because it's very difficult actually to transform plants with plasmids. You have to actually use a plasmid which is acceptable to the plant, which has protective mechanisms different to the prokaryotes. Second of all, you have to get the plasmid into the plant. So Agrobacterium has been adapted for use in genetic engineering. We've taken that this pathogenic plasmid of Agrobacterium, and genetically engineered it so that it can carry products which are useful, genes, sorry, for products which are useful. And we use the Agrobacterium to inject that plasmid into plants. It no longer has pathogenic capability. It doesn't form a tumor like this, but the plasmid can spread throughout the plant and can carry with it genes for useful products. For example, tobacco is widely used as a way of growing proteins which can be used for vaccines, for example. And those are inserted into the tobacco plants using an Agrobacterium with its attendant plasmid. Okay, so I'm sorry, this slide actually belongs a little bit before. This slide actually belongs together with our discussion about the nitrogen fixation. I'm sorry, I'll change it and repost it. But I was telling you that this nitrogen fixation by the rhizobia is an extremely complex process. But here it is being done in these little tiny root nodules extremely efficiently. And it took human beings an enormous amount of effort to learn how to take atmospheric nitrogen and actually fix it into a biologically useful form such as ammonia. We have to use huge factories in order to do this on an industrial scale to actually produce products which can then be used as fertilizers. Now think about that little tiny root nodule, and then think about the huge hunting factories that we have to set up in order to do exactly the same thing. Okay, so here's another of the Alphaproteobacteria. This one I'm including for those of you who have cats or have heard about this. This is an Alphaproteobacterium called Bartonella. a gram-negative, opportunistic human pathogen. And there are different species of it, but there's one of particular interest in health sciences, and that is this one, Bartonella henselae. And this is the causative agent of cat scratch fever or cat scratch disease. So this is actually, the Bartonella is something of an opportunistic pathogen because its normal life cycle is actually through fleas, which infest other mammals. Cats are an important vector because they pick up fleas very easily and they live in quite close association with us. But you can actually get it from dogs as well. It can infect dogs as well, but much more commonly cats. And the cats actually, in scratching at flea bites, they actually pick up the bacteria in their claws. And if you are scratched by the cat, it can then go on to infect you. Most commonly, it's pretty much self-limiting, but it is the one thing about Bartonella is it incites a very, very strong immune response. And even it often produces just a very small lesion, almost invisible sometimes. But often the first symptom that people notice is that the lymph nodes, especially under the arm or in the groin, if you've been scratched on the leg, become intensely painful and inflamed. That's called lymphadenopathy. And it's because of somewhere distally, there is an infection with Bartonella. It can actually become a bit more serious in some people, but usually it's self-limiting, but very, very easy to deal with antibiotics. It's very responsive to antibiotics. Now, this next one is called Alphaproteobacterium, is called Brucella. And Brucella is a bacterium which is forever associated with Pasteur, the great father of microbiology, the Frenchman that you've heard a lot about when we talked about the history of microbiology. The reason for that is that Brucella used to be a very important pathogen in human beings. It is an obligate parasite of mammals, and it widely infects mammals, farm animals, cows, sheep, goats, all of those animals which live in close association, and all of which especially produce milk. And animals which are in fact infected with Brucella actually tolerate it quite well often, and they will produce milk which has the Brucella in it, and that can be transmitted to people who drink the milk. It has an interesting life cycle because it actually needs to be taken up, needs to be phagocytosed by macrophages and other phagocytic white blood cells. But it's normally those white blood cells will take up pathogens and will digest them. But Brucella actually survives phagocytosis, and it breeds inside those cells. It divides successfully inside those cells and is transported to the lymph nodes and then dispersed from the lymph nodes widely through the body as a systemic infection. And the disease it causes is brucellosis, and it's also caused undulant fever, because the fever peaks spectacularly and then declines, and peaks spectacularly and then declines. And very often it's in the afternoon. People were laid low by the fever. And before antibiotics, it actually had a high mortality, especially in children and in the elderly, but in anybody who was badly infected with brucellosis could die. It's a it's a systemic disease. It infects the gut, heart, the brain, the joints, and especially in animals, it causes abortion. And this is often one of the first signs that Brucella is present in a herd, is that there is widespread abortion, but it can cause abortion in humans as well. This is why Pasteur was so famous. Pasteur developed a system of pasteurization, that is, of heating liquids up to a quite high temperature in the 70s, and keeping them there for a short time, 20 minutes or something like that, and then allowing them to cool. And that process of pasteurization was enough to kill many pathogens. It didn't kill all bacteria, it does not sterilize liquids at all, but it removes pathogens, and one of the most important of all is Brucella. And once systems of pasteurization were developed and used for pasteurizing milk, cheese, all of these products, dairy products especially, Brucellosis became much less common. It still does occur, but it is much, much less common than it used to be, and it's also responsive to antibiotics. Okay, so remember that we've already seen in the Alphaproteobacteria, we've seen two distinct systems. We saw right at the very beginning, I'll flip back here to remind you. We saw this bacterium, Azospirillum, which fixes nitrogen. We saw these, the Rhizobia, which also fix nitrogen. There are other Alphaproteobacteria, which are also very important in nitrogen cycling. And these two, Nitrosomonas and Nitrobacter, are two of them. The thing is that ammonia, which is initially produced by nitrogen fixation, is actually not necessarily the most useful biological form of nitrogen. Far more useful to plants and everything else are things like the nitrites and nitrates, especially to plants. They are very easily taken up by the root systems and are rapidly incorporated into biological molecules. These two bacteria, Nitrosomonas and Nitrobacter, are chemoautotrophs. That is, they use inorganic chemicals as a source of energy. They still use carbon dioxide as a carbon source. This is rather like photosynthesis in a way, in that they're using carbon dioxide as a carbon source, but they're using chemical energy that they get from another chemical compound to bind, as an energy source, bind those carbons together to form biological molecules. The two of them kind of interact with one another. This one, Nitrosomonas, is able to take NH4 and it will oxidize it to NO2. Sorry, that's in the wrong place. This should be down here. NO2 minus. That is nitrite. And Nitrobacter can take nitrite and oxidize it to nitrate there. Both of these, what they're doing is they're oxidizing these compounds and being reduced. Their own molecules are being reduced, so they are using the energy of, inherent in those redox reactions. They're using that energy to transform carbon dioxide into biological molecules. And the two of them look rather similar to one another. And there is a close interaction in the global ecology between all of these different Alphaproteobacteria, between those which fix nitrogen, predominantly those which fix nitrogen into the form of ammonia, and then these kinds of bacteria which will take that ammonium and oxidize it and use the energy to form nitrate nitrite, sorry, and then nitrate, which is then taken up by plants. Once they're taken up by plants and transformed into plant molecules, they can then be eaten by other things. So all of these alpha proteobacteria are extremely important in the global nitrogen cycling, in the global cycling of nitrogen. Okay, so those are the alpha proteobacteria. And what I want you to do, I want you to go and have a look on page 297 and see how to draw up a dichotomous key. A dichotomous key is one which allows you to sort out the different kinds of bacteria by asking simple yes or no questions about characteristics for each one. So if you go to page 297, you'll see how to do it. The reason I want you to do that is because it's a very useful way of putting all of this information into a form which is easy to remember. Okay, so those are the alpha proteobacteria. And for that, we'll leave that there, and then we'll proceed with our discussion on Wednesday. Please make sure that you've completed this video before then. Okay.